Yeah. Hello, everyone. I'm Abhijeet. So today I'm going to discuss about our recent research, which is malachite grain removal using algal biochar and its composite with kombucha SCOBY. It's an integrated adsorption and phycoremediation approach for remediating the malachite grain. So before starting the presentation, I would like to uh, discuss. I would like to present a proverb, a Native American proverb. This is basically, I like this proverb because it says, we do not inherit the art from our ancestor, we borrow it from our children. So basically, this is quite relevant as we are discussing about sustainability. Yeah, it's a quick background. Malachite grain is an organic compound. It belongs to the class of triaryl methane dyes. It is basically a basic dye and it carries a positive charge. Though malachite grain is banned in many developed nations, but its use is still prevalent in many developing nations due to its cheaper availability and higher efficiency in various industries. Some of the most commonly, some of the industries use malachite grain, aquaculture, pharmaceutical, food processing, textiles, and fine chemicals. So the problem with malachite grain is it can adversely impact the aquatic environment as well as the human health. It is reported to cause mutagenesis, respiratory problems, carcinogenesis, and chromosomal fractures in humans. So it needs to be remediated from the environment. For this, we have various mitigation strategies and processes such as photocatalysis, electrocoagulation, membrane filtration, phytoremediation, and adsorption. Among them, adsorption is one of the most effective and commonly implemented treatment technique due to its simplicity and efficiency in removing contaminated pollutants. So in adsorbents, like in adsorption, there are various adsorbents that has been used. For example, clay, polymers, activated carbon, nanomaterials, and biochar. Today, we'll be discussing about biochar. There are certain advantages if we use biochar because first of all, it is a cheaper adsorbent because it's produced from waste materials or byproducts of certain materials. It is quite easy to produce, and its efficiency in removing pollutants is also proven in last decades. But there are certain challenges associated to it also. To increase the adsorption capacity and removal efficiency, in majority of the case, biochar needs to be modified, and sometimes this modification can be expensive and chemical intensive. For example, in chemical activations, we use acids and alkalis, which can create secondary pollutants. And in case of physical modification or activation, we have to use higher temperature and energy, which could be energy and cost intensive. So we need to find out a methods where we can balance the trade-off and prepare efficient adsorbents. It is a well-established fact that biomass's choice affects of biochar physicochemical properties. Hence, careful consideration should be given while choosing a biomass for producing biochar. Algal biochar is different from other biochar, like biochar produced from other conventional biomasses, because algal biochar have lower surface area and carbon content, but it have a higher number of functional groups, and its inorganic content is also high, which could be very much helpful in in adsorbing pollutants. So the objective of this study is to prepare biochar from algal biomass and to undertake a novel modification so that we can improve certain aspects of the algal biochar, and hence we can remediate the algal malachite green properly. So for modification, we have used a cellulosic material. It's a SCOBY. It's a cellulosic material produced from bacterial fermentations. Many of the like the SCOBY SCOBYs, when contaminated, they generally just simply thrown out, but we can use it to modify the biochar. We hypothesized that if we modify the algal biomass with SCOBY, then the surface area and surface properties of the resultant biochar will increase significantly. And further, like instead of simply remediating the malachite green from the solution, we also use the remediated water, the partially remediated water, to grow algae in it, so that the malachite green can be completely remediated. And like for growing algae, we can reduce our dependency on fresh water. So this is an overall idea of this, what we have done. We prepared biochar, we carried out the adsorption experiments, then we also tested the reusability of biochar by performing desorption and adsorption experiments. Then we used the partially remediated water to grow microalgae in it, and we harvested the algae. And again, the microalgae which we harvested can again reuse to produce biochar. So it's basically becomes a cyclical process. So coming to the materials and method section. So as we are discussing, we have used algae and kombucha. Then we prepared a mixture, algal kombucha composite. Then we pyrolyzed the composite at 500 degrees Celsius for 60 minutes, and then we could able to produce algal kombucha biochar. Then we characterized the materials using for understanding its physicochemical properties. So as you can see from the FTR analysis, like algal biochar and algal kombucha biochar have multiple functional groups, whereas kombucha biochar do not have any functional groups. And in case of from XRD analysis, we observed that algal biochar and algal kombucha biochar are crystalline in nature, and kombucha biochar is more of a amorphous in nature. We saw like the analysis of XRD also show that AKB is having little more disorderliness in its structure. It could be due to two different materials has been combined to prepare it. Then we also perform the same analysis. So that like all the biochars are having sufficient microporous and structures. Okay. Yeah. So then the Edx analysis shown that microalgal biochar and microalgal kombucha biochar have a diverse range of elements, whereas in case of kombucha biochar, the majority of the elements is only carbon and oxygen. So we can conclude from this that algal AKB and microalgal biochar can contains various functional groups, as they have multiple elements. Then we also perform analyze the surface properties. As we hypothesized, the modification increased the total surface area, because in case of algal biochar, the surface area was 2.98 meters square per gram. But when we perform the modification, it has been increased significantly. The same is true for pore volume. The pore volume also enhanced significantly post-modification. After that, we have carried out the adsorption experiments. So for that, when first we try to study the removal efficiency by using all three biochar, algal kombucha biochar, and kombucha biochar. As you can see, kombucha biochar removal efficiency is around 20%. This could be due to, as kombucha biochar do not have any functional group, so it couldn't absorb any malachite green, whereas algal kombucha biochar and AKB are quite efficient in it. Then we studied the effect of pH. We observed like when the pH increased from 2 to 4, the removal efficiency increased significantly. This is because in highly acidic environment, the functional groups on biochar surface were protonated and positively charged. restricting the absorption of positively charged malachite green ions onto the biochar surface. Then we also analyzed the effect of initial concentration and biochar dosage. We observed if we increase the initial concentration, then the malachite green particles diffuse more rapidly to the surface, and it's increase the adsorption capacity. Whereas in case of doses, if we increase the doses, the adsorption capacity decreases because like it results in the aggregations of the adsorbent particles at the higher concentration. Then we performed the kinetic studies. We observed that algal biochar and algal kombucha biochar both fitted well to the pseudo second order kinetics. That means chemisorption plays an important role, so that like whatever the dominant mechanism could be cation exchange or wherever the involvement of chemical groups will be there. Then we also performed the adsorption isotherms. We also fitted the data with isotherm models. We have tried several several models such as Langmuir, Fredulch, Halsey, and 7 other models. But I will just try to show the results of Langmuir and Fredulch and Halsey's model. We observed that our data fitted well with Fredulch and Halsey models. It means the biochar, like the adsorption is multi-layer adsorption, and biochar is having heterogeneous adsorption sites. Then comes an interesting section. We try to integrate adsorption to phycoremediation. For this, we set 2 controls. One is positive control, one is negative control. In positive control, we have grown algae in BZ11 media along by using fresh water. In negative control, we have used algae grown with BZ11 media, but instead of fresh water, we used pure malachite green solution. And we have also used like instead of fresh water, we have used AB remediated water and AKB remediated water. So whatever, like after performing the adsorption experiments, we have utilized the water to grow in the algae. As you can see, in case of AKB remediated water and positive control, the color is almost similar. It shows that algae, like the end results is almost same to the positive control, which is if we simply grow algae in fresh water, and if we grow algae in AKB remediated water, the results are quite same. So for understanding and analyzing it, we these are the few charts which gives us an idea. Like in figure 4.a, you can see if in case of only phycoremediation, the malachite green concentration decreases from 120 ppm to around 20 ppm. But the moment when we integrate phycoremediation, adsorption and phycoremediation, the malachite green concentration decreases from 120 to almost 0. This is quite evident in removal efficiency also. The individual removal efficiency of phycoremediation, algal biochar adsorption, and algal kombucha biochar adsorption is 88, 80, and 90%. But when we integrated both the processes, the efficiency increased to 98.24%. Moreover, we also observed when you combine both the processes, the time requirement for remediating malachite green decreases significantly. As you can see, for achieving 90% of removal percentage efficiency, the time decreases significantly when we combine both the processes. The reason could be it was studied previously also that at higher dye concentrations, it impedes the algal growth, consequently affecting the removal efficiency. However, when you perform adsorption, then it will significantly reduce the malachite green concentration. Then if you grow algae in it, then it could effectively grow and remediate the dye. So we also studied the algal growth and productivity. We observed the algal productivity is highest when we are using AKB remediated water. The reason could be twofold, because algae require pH of 6.5 to 7.5 for growth, and as our biochar are generally slightly alkaline in nature, they could provide this pH range for algal growth. Moreover, like the second reason which we hypothesized, which we guessed, is like as we algal biochar have higher inorganic elements composition, so those elements can leach out into the water, and that can serve as a nutrients and other requirements, fulfill other requirements of microalgae and helps in growing it. So to conclude, the modification with kombucha SCOBY increased the adsorption capacity by threefold. This is a theoretical adsorption capacity. Then we observed chemisorption was the dominant mechanism in removing malachite green when algal biochar and algal kombucha biochar was used. Then higher algal growth was observed, and when we integrated when we used the remediated water for growing algae. And if we combine both the process, the removal efficiency, like it could almost 98%, it could remediate the malachite green from the solution. So we believe the integration of this process could be used to efficiently remove, and this type of novel modification can be environment friendly as well for remediating more pollutants of similar types. So these are the references, and this is the work which has been published in Surfaces Interfaces recently. Then at last, I would like to acknowledge to the event organizers and to my co-authors, Nageshwari Krishnamurthy, Scott X. Chang, and Balasubramaniam Paramsivam, and to the funding agencies, University of Alberta Future Energy System, University of Alberta Faculty of Graduate Studies and Research, National Institute of Technology, Rourkela, and Department of Biotechnology, Government of India. Thank you very much. I would like to discuss if you have any questions.
Yeah, thank you very much,